Okay, this is the second video capture for chapter 4, and let's go to electron microscopy. So what you're looking at here are two types of electron microscopes. On the left is a transmission EM, and on the right is a scanning EM. You cannot do both types of microscopy with one EM. There are very different kinds of setups on these electron microscopes. The pictures you're looking at of the dust mites are EMs. The one on the left is a black and white. EMs only take black and white, but you'll notice, for example, that on the pictures there are computers attached, part of the setup for the EM, and you can, as the user, tell the computer to color parts of things that you want colored, and so most EMs that you see these days have been colorized because they're more attractive, and so the dust mite on the right is obviously colored. Okay, so how different than light microscopy? Well, you're not using any light. There are no light rays. These are beams of electrons, and beams of electrons are way faster than beams of light. So you're going to get much smaller resolution. Resolving power, much better resolution. These beams of electrons have to be focused for the object, and so instead of glass lenses, which are not part of EMs, you use magnets. They are the things being used for focusing. I'll talk more about resolving power and magnification for each one as we get to them. Okay, so let's talk transmission EM. There are different ways to prepare a specimen for transmission. You can coat, well, let me start at the beginning. Let me take you through this picture over here on the right. Yep, okay. Alright, so notice the magnets, which you see as gray objects. Notice a specimen right there. There is where your specimen is. So in other words, the beam of electrons coming from right here is focused to go right through a thin specimen, and eventually it hits a screen where it's then turned into information for the computer. Now, this requires that the specimen be cut very thin. And when I mean cut thin, I mean out of the realm of cutting it with a human hand, because you can't get it thin enough. Now, when it's cut this thing thin, it would be comparable to you cutting a loaf of French bread using a serrated knife and cutting it as thin as you could possibly get, and then lifting a piece right out of the middle of the bread. That thin. Well, much thinner, obviously. Now, the way you prepare the specimen can vary. Sometimes they take this specimen that's been cut thin and they coat it with heavy metals, like maybe gold, uranium, lead, things like that. Sometimes they spray materials on the specimen. Sometimes the specimen is frozen. Sometimes they do a process called shadowing, and actually there are two pictures over here, and this one at the bottom right there shows shadowing. There's your bacterial cell outline right there. This is an endospore, which we've already talked about. Let me erase that. And notice right here it looks like there's a shadow on one side. This is done by shadowing. Now the picture up here has not been shadowed. It's also a spore inside of a cell, but it gives you a very different view. Compare these EMs with a gram stain showing an endospore in the left-hand cell and a phase contrast view where the spores are refractal and light. They look like tennis rackets. Well, they look like the racket part of the tennis racket. The transmission EM can get down to about or less than 1 nanometer. Think about that. A nanometer is 10 to the minus ninth meter. A billionth of a meter. And the magnification can be as high as a million times magnification. Now remember, your microscope only magnifies 1,000 times. So we're talking 1,000 times greater magnification here. Advantages, disadvantages. Well, I just gave you the advantages. There are some disadvantages. In order to prepare specimens for any kind of EM, whether it's scanning or it's transmission, the specimen is put into a vacuum. That means that you have to dehydrate the specimen. Otherwise, in a vacuum, the water inside the cell would vaporize and destroy everything. Which means that then your specimen is totally dead. You're looking at dead things. Obviously, you can use a light microscope to look at live things, so that's a limitation. Also, the TEM is way larger than a scanning EM, and you really need to know what you're doing because it's much more complicated than a scanning EM. Okay, so let's go to, let's see, I think I got a couple of pictures here. Here's what's called freeze etching of a specimen, and then it's covered with metal vapors, giving you a kind of a shadow, which you see on the far right. And then at the bottom you see some examples of TEMs. You've got a cell on the left with some, looks like inclusions in green. This is obviously these have been colorized. And then the cell on the right, you're actually looking at an entire cell in cross-section. The orange is the cell wall, and those blue things inside of the cell are, I believe they're gas inclusions inside there. Okay, so let's go to the scanning EM, which is a totally different kettle of fish here. Okay, so in this scenario, here is your electron gun up here. You see the magnets. Now look where the specimen is down here. So the electron beam comes through here and it hits your specimen and it knocks electrons off, like dropping a container of water onto an object on the ground, and when the water hits that object, the water molecules spray off in different directions, and these electron sprays are captured by the information to give you an image. Now the picture you're looking at the bottom, you see, is a head of a pencil. This is pencil lead, but right here, magnified even higher, you can start to see bacteria, and here even higher. So magnification on a scanning EM up to 200,000 times magnification. So remember, TEM was upwards of 1 million or more, so we're talking 5 times greater for transmission magnification compared to SEM. And as for resolution and resolving power, you can get down to about 5 nanometers, whereas with a transmission EM you could get down to 1 nanometer or less. So you're looking at a resolving power of 5 times smaller with a TEM compared to an SEM. But again, both of these will allow you to see bacteria very nicely. Just depends on what you want to see. The TEM, you're looking at things internally because you're looking at a thin slice. Now remember my French bread analogy? In the case of a scanning EM, if you put the French bread, the whole bread, not cut, French bread whole, down on the floor, and or in a EM, and you then take a picture of it in the scanning EM, you would get a 3-dimensional view external of the bread. You're looking at the external architecture, and that's what you're using the SEM for. And that's what you're looking at on the pencil head. It's also what you're looking at right over here, which is a bunch of yeast cells. So this is not used for internal, it's used for external. It's much easier to use. In fact, we have a scanning EM that they use down in the tech area where they're doing things like wafers for computers and things like that for TI. TI has some of the scanning EMs also. Again, I've already given you the disadvantages. And again, advantages, if you only want external view and you want something easy to use, easier, then a scanning EM is much better. Here's a view of a hypodermic needle with blood cells on it. End of a hypodermic needle using scanning EM. And different views of transmission. Whoops, wait a minute. Let me go back to that. Okay. Here is TEM. So in your book, you'll see references to TEM and SEM. That's what it means. This is a TEM over here. This is a scanning EM. This is a scanning EM. Red blood cells. These are sickle cells, by the way, in green. Here's a good example of how adding color can really give you much more information. The computer was told to colorize the cells that were stretched out to a certain diameter more than a regular red blood cell, like sickle cells are, in green, so you can differentiate them easily. This is a scanning EM of a paramecium. This guy right here is a regular light bright field type of microscopy for comparison. Okay, let's go over to staining. Forget EM, because we don't have an EM here, and we're not going to be doing EM. Some large universities have them, and sometimes they offer courses in electron microscopy, but we're talking light microscopy in this course here. Alright, so you already know what it is. We use salts with charges on them, plus and minus charges. One of the salts is, one of the ions is colored. It's called a chromophore. And they can be charged positive, they can be charged negative. If they're negatively charged, they're called acidic dyes, and if they're positive, they're basic dyes. I have an asterisk here, mainly because that's the group that we use all the time in lab. For the gram stain, for the acid fast, everything we're doing is basic dyes. So you've used methylene blue, you may have used crystal violet, saffronin. We use bunches of dyes, probably 10 different dyes in this class. They're all basic dyes. Basic dyes, having a positive charge, are attracted to the cell wall of bacteria, which have a negative charge. So the dyes attach to the outside of the cell, usually, not always. The view that you're looking at in the middle here in blue is a capsule stain, which I showed you before. And notice that there's a dye in the background. There's actually two dyes. There's one dye that's dyeing the bacterial cell a deep, deep purple inside of the capsule, which is clear. And then there's a dye in the background, and that background dye is an acidic dye. It's repelled by the cell wall because they both have the same negative charge, whereas the basic dye is dyeing the bacterial cell itself. Okay, so you can look at things as a wet mount, and you can look at things as a stain. Depends on what your aim is. If you just want to see them, see if they're bacteria in blood, you want to do quantitative counts, you want to see shapes, you want to see tiny little microorganisms, then you're going to be doing staining. Bacteria are too small to look at effectively on wet mounts. Now we do it one time this semester, and after that you'll really get to appreciate staining, because staining gives you much better elucidation of bacteria. Now if we were looking at big guys like algae, we wouldn't stain them, we would do wet mounts of them because they're big and they're colored green. The other advantage of a wet mount is that it allows you to see behavior of the organism. Are they motile? What type of motility? Things like that. Simple stains versus differential stains. In order to stain, you have to make a smear, and you can see a smear right over here. It's on a glass microscope slide, obviously, and it's already been stained. That's why it's blue. But that's your bacterial smear. If you only use one dye, you've got a simple stain. That's what's been done over there on the right using probably crystal violet. Everything will be the same color. You could have 10 different species of bacteria. You could have algae. You could have everything mixed together of different organisms, and everything would still be the same color. Now, sometimes simple stains are good. If you just want to see if there are bacteria there, simple staining is nice because it only requires one step. You stain it, you wash it, you look at it. But if you want to get any further information about your organisms, you want to see anything about differences in the way they stain or their structure or anything like that, peculiar or specific things inside of the cell, then you want to use multiple dyes. And that's what a differential stain is. It uses 2 or more dyes. We actually will be doing 3 different differential stains in lab. We do the gram stain, we do the spore stain, we do an acid-fast stain. We will also be looking at prepared bought smears of flagella and capsule stains. Now, you can use differential stains to do a lot of things. You can use stains for staining the wall. That's what a spore stain and a gram stain are used for. You can stain flagella, things outside of the cell, or you can stain things inside of the cell. So, for example, the shot you're looking at right here, this is a stain for metachromatic granules. They are phosphate inclusions inside the cell. We talked about that before in Chapter 3. And they look like little blue dots inside of the cells. And there's only one genus of organism that makes these phosphate inclusions, Corynebacterium. So this is a very important stain for that particular organism. There's a spore stain over here on the right. You can see the green spores inside the cells. And over here is a flagella stain. And you can just make out maybe some little flagella attached to the bacterial cells. Notice that the acid-fast stain and gram stain were developed in the 1880s. These guys have been around for a while. Okay, so I've already introduced you to the gram stain, but now I want to get into the mechanics of it. We're going to be doing this umpteen times in lab, so you'll get a lot of experience. Note the gram stain up at the top right. You're looking at it on oil immersion. You've got 2 different kinds of bacteria there. You've got coccus-shaped clusters of blue cells. Those are your gram-positive that retain the purple dye, crystal violet. And then you've got some small rods, which are actually 2-3 times as big as the cocci in length, but they are rod or bacillus shaped. And they are pink, and they decolorize and pick up the safranin, which is a pink dye. Most people immediately jump onto the crystal violet gram-positive because the purple jumps out at you, but the pink color is the gram-negative, and they're just as valid as the gram-positive. So, you see 4 major steps here. Okay, so let's go back in time to when these 2 bacteria, for example, E. coli and Staphylococcus, were mixed on a slide together, because obviously they were mixed together on a smear here, because it's a mixed smear of 2 bacteria. Now remember, we've just made the smear, we let it air dry, we heat fix it so that the smear stays on the slide and doesn't come off, and now we're ready to stain. Okay, so first we're going to use crystal violet, the primary dye. Now remember, we've got 2 bacteria mixed together. If, for any reason, you stopped the gram stain right there, after staining for a minute, and you wash it off with water, and you look at it on a microscope, all right, everything would be purple. Everything would be purple. Both the gram-positive and the gram-negative. Crystal violet goes into the peptidoglycan layer, and the area between the cell membrane and the PG in the wall. It goes into both gram-positive and gram-negative. Everything will be the same color. Okay, but obviously you don't do that. You don't stop it. Okay, so we would normally go on, trying to get that out. Okay, so normally we would go on, and we would now add the mordant, which is iodine. And the job of the mordant is not to give any color variation. It's to intensify the binding between the crystal violet and the cell wall. Iodine is the color of freshly made dark tea. It is not a dye, is not a dye, is not a dye. So again, if we stop the gram stain right here and look at it in a microscope, everything is still purple. Everything. Okay, but obviously we don't do that. All right, so here is the make it or break it step right there. The next one is the decolorizer. All right, you can use alcohol, you can use acetone. We use a 50-50 alcohol-acetone mixture. We only decolorize for about 5 to 8 seconds usually. Now that time is critical because in that time period, the decolorizer will remove the crystal violet from the gram-negative, but it's not on there long enough to remove the crystal violet from the gram-positive. The gram-positive retain the crystal violet, the gram-negative lose the crystal violet. So, if you stopped it there and looked at a microscope, gram-positives would be purple, and what would the gram-negatives be? No color. They're clear. Crystal violet's been leached out. Now that then tells you why you have to add one more reagent, safranin, which is a pink dye. It goes into these clear bacterial cells and turns them pink. Safranin is a basic dye, just like crystal violet. It binds in that PG layer. But safranin has no effect on the gram-positives because gram-positives still have crystal violet. They stay the same purple. Now, you can mess this up in different ways. You can leave decolorizer on too long. That's probably the number one way that people screw things up. Or they don't leave the decolorizer on long enough, and too much crystal violet stays in the cell wall and it can't be stained effectively with the safranin. Also, some bacteria will spontaneously decolorize. The genus Bacillus tends to do that. So when you look at them, even if you've done a perfect gram stain procedure, you'll see some pink and you'll see some purple. It's not anything you've done, it's just that particular organism. Also, old bacteria, that is days old, tend to not stain well, and so you get erratic staining and errors in staining as a result of that. Okay, so let's switch gears to classification. Okay, we're in the area called taxonomy. This is the study of classification. It was started by Carolus Linnaeus, the father of taxonomy, 1700s. I already mentioned he came up with the first two kingdoms, plant and animal. He actually came up with a third one called Infusaria, which had all the little microscopic things, but that never caught on. Anyway, so he's got another claim to fame, and that is he came up with a way of naming these things. It's a two-name naming system called binomial nomenclature. Binomial, two names, nom, name. Nomenclature, a naming system. So you see over here, genus, first name, species, second name. Notice that it is italicized, or the other way to do it is underline. So in your textbook you'll see them, genus and species names always italicized. If you were going to, in the old days when we had typewriters, if you were writing a paper with genus and species names, you had to underline. There was no italicizing with a typewriter. Okay, the names. People really object to these names of organisms. Humans are Homo sapiens. The domestic cat is Felis domestica. I mean, those are pretty easy. But you throw Streptococcus pyogenes at somebody, and all of a sudden they freak out. I don't know why. It's not that much more different than the house cat name of Felis domestica. But if you work with the words, they sometimes tell you something. For example, pneumonia, the species name. By the way, look, notice that species is not capitalized, only genus. All right, pneumonia tells you that this organism causes pneumonia. Strep pyogenes, which is the cause of strep throat, causes a pyogenic infection. Pyogenic, pyo means pus, genic means to make, pyogenic means makes pus. So that tells you something about the problem, about the disorder. Now, often people's names are in the genus and species names, usually genus, like Klebs, his name is in there, and Escherick, his name is in there. Sometimes you'll see something about location of where the organism lives, like coli for colon, because E. coli lives in your large intestine. Or sometimes you'll see something about what the organism is. Remember the field called mycology? Myc means fungi. This is a fungus right there. And sac, as in saccharin or saccharolytic, pertains to sugar. So this is a sugar fungus. And let's go for the species here for a minute. You know this word, a variation of the word. The variation you probably know is cerveza, yes, which means beer. So this is the sugar fungus that makes beer. See how easy this is? Sometimes shape of an organism, coccus, round. Arrangement, strep in chains. Okay, the guy down here at the bottom, Entamoeba histolytica, lytic, breaks up. Histo, those of you from A&P, think about histology, the study of tissues. So histolytica means destroys tissues. Amoeba tells you that it's a protozoan, an amoeba. And enta refers to intestinal, so it's an intestinal amoeba that destroys tissues. Okay. The other view you're looking at down here on the right is the evolutionary picture of the various kingdoms. And again, I wanted to point out your bacteria are down here at the bottom. Prokaryotic cells preceded eukaryotic by a couple of billion years. Okay. So, going forward with that, here's a look at time. Here is origin of the Earth right there. Okay, 4.6 billion. Here's the development of bacteria. And we are talking somewhere between 3 and 4 billion years. The first bacteria were anaerobic. Look at the word anoxic, meaning no oxygen, because there wasn't any. They were using things like sulfur and phosphorus and all sorts of various elements that were in the atmosphere, inland, so on and so forth. Now, at about 2.5 million years ago or more, somewhere in there, we have the development. Actually, let's go back up into this time, further back. We start to get the non-oxygen producing photosynthesizing bacteria. These are photosynthetic bacteria right here, but the original photosynthetic bacteria did not use oxygen. Because remember, oxygen didn't come along until a little bit later. So then if you go forward up into this period of time, now there's oxygen, and now we have the photosynthetic bacteria that do produce oxygen. Then, as we move up to about right here, somewhere around 2 billion, they've identified fossilized eukaryotic cells back to about 2 million. We have eukaryotic cells come on the scene. The views over on the right, this is Australia, although you find them in other places, but they're most preserved in Australia. This is a picture of Australia right here. You're right off the shore. And these guys right here that look like coral are not. They are fossilized bacterial aggregates, as if you took, I don't know, layers of stuff, you know, like meat. Like, let's say you take a packet of bologna and you leave it in your refrigerator for, you know, a ton. You know, it comes sliced. But over time it compresses and becomes solidified into spoiled material. That's basically what you got over here. Well, it's not spoiled, but it's fossilized. In other words, it's hard, and it's layer and layer and layer and layer of bacteria, bacteria. And they are hardened things called stromatolites. And you see some more stromatolites over here under the water. So here's a look at domains. You're familiar with kingdom, maybe not so much with domains. Domains are now the biggest categories of organisms, include not just microorganisms, but organisms. So these are bigger categories than kingdoms. The three domains are the eubacteria, that is true bacteria, like E. coli, staph, the ones you know. The archaea bacteria, which are the what are sometimes called extremophiles. We'll talk a little bit about them more. Extremophiles live in extreme environmental conditions like high salt, high heat, high acid, and other things. So we have two domains of bacteria. You see the eubacteria right here. You see the archaea right there. And then this big domain over here is full of the things that you know, cats, beetles, mosquitoes, all the eukaryotic guys are over in this area. Okay, so what I was trying to show you here was going from domain down to species, what this looks like. And what I did is I have humans over on the left. Wait a minute, I'm trying to get rid of the bar. Oh. Okay, here is a human, and here is E. coli. Okay, so we have two different domains, eukarya versus eubacteria. Kingdoms, we're in the animal kingdom. Bacteria, I'll just call them bacteria kingdom. Then we've got phylum, subphylum, class, and so on. You don't need to know all this. Many of these taxonomic ranks, like orders and families and classes, you don't actually even find in the bacteria. By the way, these rankings, kingdom, phylum, subphylum, class, subclass, order, suborder, family, subfamily, are called taxons. It's the name for that level. And then we finally get down to genus and species. Escherichia coli, Homo sapiens. Now, in the case of bacteria and other microorganisms, you can even get down to something smaller than a species. They're called strains or serotypes. So, for example, there is a strain of E. coli called 0157, which is a super pathogenic E. coli. It's the one that's been associated with undercooked hamburger, Jack in the Boxes, on raw spinach. They found it lots of different places. Excreted in feces, contaminates food, people die, blah, blah, blah. All right, so that's a look at taxonomic rankings. Okay, so how do you classify them? We're going to be doing an unknown in the micro lab, and we use various ways to classify bacteria. We look at physical characteristics like staining. We do gram staining, things like that. We look at how they grow on agar plates and how they grow in tubes of different kinds of media. We run biochemical tests to see if they use certain sugars, or do they make an acid from a particular sugar, or do they use a particular amino acid, or can they use gelatin. These are called biochemical tests. We also will be doing some serological testing, which is where we use antibody to a particular organism to identify an organism. The little thing you're looking at right here, oops, wait a minute, let me go back. Okay. Whoops. This thing right here, this little cartridge right here has an antibody inside of it. It's very similar to a pregnancy test kit, by the way. And they are testing for heartworm. This is a common way that heartworm is identified now. using a test kit. So that's a serological test. You can also classify organisms based on viruses that infect them, because bacterial viruses are very, very specific for their host. So, for example, there might be one particular virus called a phage that infects E. coli 0157, but other E. coli viruses would not have infected at all, even though there are other E. coli viruses. It's just that this particular phage would be specific for 0157. And that's what you're looking at right here. There's a bacterium growing in the background in green, and these little openings right here are areas of destruction caused by a particular virus. So you use a known virus to infect the bacterium, and whichever type of virus infects the bacterium, it tells you something specific about the bacterium that you have. You can also analyze nucleic acids and proteins, DNA, enzymes, orders of amino acids, all sorts of things like that to get more information about the organism. Okay, taxonomic keys. We're going to be using them in lab. Here's an example. For example, in this key, your first choice up at the top, it's asking you, is it gram-positive? Yes, no. If it is yes, then you go to the right. If it's not gram-positive, if it's pink, which means it's gram-negative, you go to the left. It's a no. Then it asks you, is it rod-shaped? Yes, no. Let's say that it is rod-shaped. You go off to the right. Can it grow in the presence of oxygen? Yes, no. Yes, you go off to the right. Does it use lactose? Yes, you keep going to the right. Eventually, you end up getting down at a genus, in this case, Escherichia. So at every turn, you have two choices. That's why this is called dichotomous. Two choices. And this is called a key. Over on the right, you see another way to do taxonomy. This is called numerical taxonomy, where you assign points for each feature of a microorganism, and then you put all the information into a computer, and the computer tells you what organism this organism is most similar to based on a numbering system. We're not going to be using that type of classification. And that is it.